So today I want to say some more about innate immunity, particularly kind of the cellular innate immune response. So I'm going to talk through that. I'm also going to talk a little bit about some kind of big picture stuff with regard to comparative immunology, evolution of the immune system, although I'm only going to say a little bit about that. I also do have some data from a paper that would have been part of that literature assignment if I had it, because I want to tell you just a little bit about the story of those data. I don't know if we're going to totally have time, and I really had trouble figuring out the order of the slides for that part to make sense, so I just put it at the end. So it might feel like an abrupt change. Sorry. So where we left off last time was talking about the different groups of pattern recognition receptors. And the goal today is to talk through some of these specific pattern recognition receptors and discuss details of what those receptors are binding and kind of some of the unique aspects about those receptors. One thing that I want to remind you that we talked about a bit last time and is going to come up today as well is that last time we talked about sort of the general kind of process of inflammation, which I said often happens in response to bacteria, although that is an oversimplification. And that is the one that leads to the production of cytokines like TNF-alpha, IL-6, IL-1 beta, as well as a lot of chemokines. And so we saw that process. This is the one that gives you the redness and the swelling and that's that sort of classic inflammation process that we talked about. So recall this. Also recall that we talked about the antiviral response. Obviously the antiviral response is generally happening in response to viruses, though again, that's a bit of an oversimplification. And for our purposes, we're going to say that that leads to the production of type 1 interferon, which is interferon alpha and beta. And those can eventually lead to the production of ISGs. So this framework is going to come back up today. So here it is for our reference. And part of the reason why it's important to talk about this is because when we talk about some of these groups of PRRs, we might classify them as doing more of one or the other of these processes. It's actually a big part of how we're going to classify them. The kind of beginning of our understanding of PRRs was this discovery of first the Drosophila protein Tol, and then these similar proteins in humans and other mammals, the Tol-like receptors. And so the first one of these that was found was Tol, but now we know there's a whole family of Tol-like receptors. These proteins have a domain that allows them to recognize a microbe. You can see that domain here. That domain has a structure called a leucine-rich repeat, LRR. I'll say a couple things about leucine-rich repeats in other locations elsewhere today. So this is the LRR. And you can see there's also a signaling domain. And I told you about the discovery of Tol last time. When we think about TLRs, we can divide them into two major groups of TLRs. We now know of about 13 TLRs in human, slightly different numbers in other organisms. And one of the ways that we can group those TLRs is we can realize that they actually live in one of two different locations in the cell. So some of them live on the plasma membrane. Others live inside the endosome. So the ones that are on the plasma membrane can detect microbes that are outside of cells. You can see that as an example here. While the ones that are in the endosome can detect things that maybe have come in by phagocytosis or are in the endocytic pathway. So one way that we divide up these TLRs is whether they are at the plasma membrane or the endosome. And if you look at this table from your textbook, you can see plasma membrane group, endosome group is one of the big kind of dividing factors. So that's one way we can think about dividing these up. If we look at them, we can also see that they sort of divide up nicely in another way. You can see that one set of them tends to bind to lipids, and another set of them tends to bind to nucleic acid. This one right here that binds to a protein, I'm going to throw in with the lipids. So we've got sort of nucleic acid binders and lipid protein binders. It turns out those match up with the locations on the cell. So these lipid and protein binders are the ones that are on the plasma membrane. And these nucleic acid binders are the ones that are in the endosome. So again, we can divide up our TLRs really nicely into two groups. And you can kind of see this here as well, where we've got our plasma membrane, ones that are largely looking at things like lipopeptides, LPS, different types of lipids, etc. If you think about LPS, which is part of the cell wall of a gram-negative bacteria, as an example, that was actually the first PRR that, part of the toll-like receptors that we understood, was the one that binds to LPS. If you think of LPS as an example, what kind of microbe, I kind of already told you this, so don't overthink it. What kind of microbe is LPS going to be on? Yeah, Remy? Bacteria, right? So ideally, we would love it if the cell said, oh my gosh, it's bacteria. I should make a bacteria type response, right? If you look at this little diagram, again, that's slightly oversimplifying things, where is the cell most likely to encounter bacteria? Yep, Demir. Yeah, the bacteria are often found outside of our cells. And so they're probably going to be detected by a plasma membrane receptor. Viruses like to be inside our cells. Pretty much the only thing that can be detected from them is nucleic acid. And these receptors inside the cells, which happen to be the ones that look for nucleic acid, tend to be recognizing viruses. As I have already told you, one of the key things that all of our PRRs can do is lead to changes in transcription and different cytokines being produced. But the specific details of the magic arrows on this slide, in terms of the transcription, can vary with the different PRRs. I've introduced you to these two groups of toll-like receptors. I said one of them's at the plasma membrane. It's detecting things like lipids from bacteria. It's probably going to detect bacteria because of its location. One of them's inside the cell. It's going to be mostly detecting virus products. I've kind of set this up for you. And it turns out that these two groups lead to different types of transcription. And so that's kind of how we regulate this response differently. So some PRRs lead to activation of this transcription factor, NF-kappa B. Everybody wants the B to be a beta. I don't know how many people have called it NF-kappa beta. It's actually a B. Yes, it does stand for something. We don't need to worry about it. But NF-kappa B is this one transcription factor. So one option downstream of PRRs is that we can turn on this transcription factor, NF-kappa B. When NF-kappa B is turned on, it allows for the transcription of cytokines like TNF, IL-1 beta, IL-6, chemokines, and some other important proteins that we'll hear about later in the semester. And in fact, it will allow us to make that antibacterial response. So NF-kappa B basically is this transcription factor that's controlling this side of the response. With some other PRRs, we can get a different kind of transcription factor activated. That transcription factor is called IRF. There are different IRFs. There's different NF-kappa Bs too. IRF stands for Interferon Regulatory Factor. So guess what gene interferon regulatory factor turns on? What is it? Interferon. So we could alternatively have this IRF transcription factor that's going to turn on our type 1 interferons. And so our different PRRs can predominantly induce either this type of sort of acute inflammation response or the antiviral response based on which transcription factor they signal to. So, if you think about our plasma membrane toll-like receptors versus our endosomal toll-like receptors, I'm going to have you make a hypothesis that we will then see later slides to see if you were right or not. What transcription factor would you predict that this toll-like receptor, here it's TLR4, what transcription factor would you expect TLR4 turns on? Yep, Jameer. NF-kappa B, because we want to make a response that'll work well for bacteria, and that's kind of what we think of as the bacterial response. What type, and that's generally going to be true of a TLR that binds to, say, one of those lipids. What type of transcription factor do you think TLR3 or another one of these endosomal TLRs that binds nucleic acids will turn on? Carney? IRF. So let's see. Oh my gosh, cell surface toll-like receptors induce NF-kappa B, exactly as Jameer predicted. So here you can see our cell surface toll-like receptors. This is specifically an example of TLR4 binding LPS, but I'm not going to go crazy into those details. It leads to some signal transduction, and that results in our NF-kappa B transcription factor making the cytokines that we've talked about. This can be the thing that happens when we recognize LPS, or when we recognize flagellin, or a lot of different important bacterial products. Alternatively, Carney predicted that the endosomal TLRs would signal using IRFs. Oh my gosh, also correct. So here again, we can see our endosomal TLRs. In the case of endosomal TLRs, they're always binding some form of nucleic acid, which is often thought of as being indicative of a virus. They're leading to some signal transduction and eventually turning on a transcription factor of the IRF family. Yes, this shows different IRFs. We're just going to call them all IRF now. I'm not going to distinguish 3 versus 7 like this figure does. I'm just going to say IRF. So you can see that we can kind of get this nice distinction where different types of microbes could signal to different PRRs, and that could allow us to make an appropriate type of response. So it all ties together. After the TLRs were described, some other types of pattern recognition receptors were described. In general, they have families. There's not just one member. We see multiple proteins that do similar things. I'm not going to talk about these. I'm actually going to move on to these, which are the RLRs. And the RLRs stands for RIG-I-like receptors. I'm going to show you some information about the RIG-I-like receptors on the next slide, but I want to point out a couple of details about RIG-I-like receptors from this slide, and again, ask you to make a prediction. So the RIG-I-like receptors tend to be in the cytoplasm. That's different than what we've seen before, where we were largely seeing plasma membrane and endosomes. So this is a good thing, because now we are doing surveillance in all the parts of the cell. It's not like there's some part of the cell where a microbe can slip by, because we're going to be looking everywhere. We're now looking in the cytoplasm. And the thing that RLRs are going to look for is RNA. Particularly some weird types of RNA that are made during viral infection. That's why it says viral RNA here. So, or I'll stand in front of it. You definitely can't see anything now. If you have to guess, if you have to make a prediction, when we see signaling through RLRs, which of these two pathways do you think RLRs might signal through, and why? Michael.
IRF, because it's responding to a virus and produces an Interferon response.
Right, so again, we're seeing a response to nucleic acids. And usually when we're thinking about nucleic acids, we're thinking about a response to a virus. And in particular, I told you that these are responding to a type of RNA that's found in viruses. So you'd hope that they signal through IRF and give us an antiviral response. That would be ideal for this to make sense. Oh my gosh, they signal through IRFs. And lead to the interferon response. And so yes, on this and other slides, you can see that there are other components of the signaling cascade that are important here. But what we're going to care about is who's the PRR, who's the transcription factor in most cases. I will say more about, maybe, depending on time, SARS-CoV-2 later. But a member of the RLR family, specifically a member named MDA5, is super important in SARS-CoV-2 responses. So then we can move on to another group. So TLRs stood for toll-like receptors, because the first protein in this group that we found was toll. And then we found a bunch of similar proteins. Then we learned about RLRs. The first member of this family that was found was RIG-I. And then we found a bunch of similar family members. The next group is going to be called the NLRs. And you can imagine that it should be N-like receptors, right? That's what you'd like it to be. Yeah, it's not. NLR currently stands for Nucleotide Binding Domain Leucine-Rich Repeat Proteins. The reason for that is because it was actually first named after this protein called NOD. So they were the NOD-like receptors. And then we realized that NOD was super weird and it wasn't actually like any of the rest of them. And we were like, oh, that name kind of sucks if none of them are actually like NOD. So we're going to keep NLR and make up new things it stands for. So this was like someone trying to make up a thing that was true that also went with NLR. So yeah, it's super weird. But it does tell us some interesting things about the NLRs. One of the things it tells us is that NLRs have leucine-rich repeats, and you can see the leucine-rich repeats drawn here. Have you ever heard of leucine-rich repeats before? Anyone? Michael? Yeah, that was the part of the toll-like receptor I pointed out. So you're seeing maybe some similarities in similar domains being used for innate immunity. They also have this domain called an NBD. And then they have an additional domain, and it can vary depending on the NLR. This allows for protein-protein interactions. So there are some NLRs that are called like NLRP1. That means that here they've got the P kind of domain, and they were the first one. There's also NLRP2 and NLRP3. And there's some that are like NLRC4, because it has the C kind of domain and whatnot. But there are a couple of, and so this is kind of big picture, understanding the NLRs. There are two kinds of signaling that we can see with the NLRs. One of those kinds of signaling is that NLRs can signal through NF-kappa B and give you our nice classic inflammatory response process. And so that is one thing that some NLRs can do. But that's not actually what NLRs tend to be famous for. NLRs tend to be famous for something else. And there are two other things I want to talk about on this slide to help you understand both what I'm going to tell you NLRs are often famous for, as well as some other thing that we're going to move into going forward. So here are the NLRs. Here are two other proteins that are not NLRs that are shown for comparison. So first, let's look at the top protein. And you can see the general structure of this top protein compared to these NLRs. What do you notice if you compare the structure of this protein with the NLR structures? Yep, Jameer.
The effector domain is the only thing that's different?
Yeah, so basically, we've got an NBD that looks just like what we had in the NLRs. We've got an LRR, which looks just like what we had. The only difference is this effector domain, and I don't know, variation in effector domains is already something we see. So this looks pretty similar to an NLR, right? Turns out that this is one of the proteins that make up the plant immune system. So while animals are using this kind of protein structure as a big part of our innate immune system, plants basically use an almost identical kind of protein structure for a lot of their immune responses. So it's kind of cool, and I'm going to say a little bit more about plants and kind of immunity across kingdoms later. So that's kind of cool. Then we've got this other protein. Let's look at what do you see that's in terms of similarities with this other protein? And they are drawing things specific ways for a reason here. So like If you notice something based on the drawing, they drew it that way on purpose. Go for it.
You're saying the similarities
Between this one and the NLRs.
it has the same domain and bonding?
Okay, so we've got the NBD. We've got a card domain, which is one of the choices of effector domains for the NLRs. What else do we have?
Well, it looks like a leucine protein.
So it's not an LRR, it's a WD-40, but structurally it's really similar to an LRR. Look, they even drew it basically exactly the same. So this is another protein that looks really similar to an NLR. It's called APAF1. Any of you remember ever learning about APAF1? You did, but you might not remember it, like, because it's not focused on hard when you learned about it before. So APAF1 was a protein that came up in Bio-250. APAF1 is involved in the apoptosome, or as Dr. Dunaway likes to talk about, the pinwheel of death. So with the apoptosome, APAF1 gets brought together. It actually gets brought together partially by WD-40s and turns on caspases to allow for apoptosis in terms of cell death. And what you'll notice is that this protein, APAF1, that's involved in this type of cell death apoptosis looks pretty similar to the NLRs. The thing that the NLRs are particularly famous for is that they also form a structure that looks just like the apoptosome. However, the structure is not part of apoptosis. Instead, this structure, which involves a whole lot of NLRs coming together with an adapter. You can think of the adapter as taking the exact same place as cytochrome C. And an unactive caspase. In this case, it's procaspase 1, whereas in the apoptosome, it's procaspase 9. And make this pinwheel-y structure. But it's not, like I said, involved in apoptosis. It's actually involved in inflammation. And so instead of calling it the apoptosome, we call it the inflammasome. The goal of the inflammasome is, again, to turn on some caspases, particularly caspase 1, while it was caspase 9 and caspase 3 in the case of the apoptosome. And one part of the inflammasome is that some cytokines need to be cleaved by a caspase to be active. So part of what the inflammasome can do is that the inflammasome can process some cytokines. And so NLRs are often really important for inducing an inflammasome, which can allow for the processing of cytokines, particularly IL-1 beta. There is a second thing that happens with the inflammasome. And in order to tell you about this second thing, I have to go back for another Bio-250 business. So when you took Bio-250, you basically learned this, which were some forms of cell death. You learned about apoptosis, which you were taught as programmed cell death. And you learned about necrosis, which was kind of like accidental cell death. The cell just blows up. And in apoptosis, the cell undergoes this programmed process. Really, the phrase programmed cell death means that there was some kind of signaling for that cell death. The cell died on purpose because of signaling. Whereas with this necrosis, the idea was the cell died almost on accident. we now know there are more kinds of programmed cell death than just apoptosis. So there are other kinds of signaling that lead to apoptosis. When I, as an immunologist, think about apoptosis, I think about some really key events. So in apoptosis, the cell is going to fragment its DNA, and it's going to make these little membrane blebs. It's going to make like these little compartments and little pieces of itself that are going to go off and maybe get eaten by a macrophage. The way I think about apoptosis is that apoptosis is really quiet cell death. It's a cell that's just going to fold itself up and die and not disturb any of its neighbors and not make any problems. It's sort of how I think about apoptosis. For example, you had a whole bunch of apoptosis happen during development. At one point during development, you actually have webbed toes and feet, and then there's apoptosis where all the cells in the webs die. And those cells are just like nicely, quietly dying and not messing with anything else, and that's the end. So there's another type of programmed cell death. More and more keep getting discovered. I'm only going to say about this one other one. It's called pyroptosis. And you can see the word pyroptosis here. When you look at the word pyroptosis, apoptosis actually has some roots that are related to like the falling of a leaf. What does pyroptosis make you think of? Just looking at the word. Yeah, Michael. Burning, pyro. Pyro means fire. Pyroptosis is fiery cell death. And with pyroptosis, we are going to have some signaling, and then the cell is not going to shrink up and put all of its stuff in little compartments and hide them. This cell is going to blow up spectacularly. It is not quiet. It is that cell spewing out all of its intracellular contents and definitely disturbing its neighbors. And you can imagine that could be useful in, say, the case of an infection, because this cell can be saying, hey neighbors, there's something bad around, I'm going to die, but all of you get ready. In terms of giving a message. So we have this, it really does look like programmed necrosis. And our NLRs also can lead to pyroptosis. So the inflammasome can actually lead to making a pore that allows for our cells to undergo pyroptosis. So NLRs both lead to some cytokine production, and they also lead to inflammasomes and pyroptosis. And you can sort of see that there's a protein that looks like an NLR structurally, and that leads to a different kind of cell death, apoptosis, through a similar kind of structure. So just to give you a little context, these are some things that have been shown to be recognized by different types of NLRs. In fact, most of these people have said are recognized by this one kind of NLR, NLRP3. And you can see we've got ATP, cholesterol, urate, glucose, amyloid beta, alum, asbestos, silica, UV radiation, skin irritants, and a number of different microbial products. And so you can see how how inflammation downstream of these NLRs can be related to a lot of different clinical conditions. This also tells us there's some additional signaling work that we are not clear about. And if we think about a lot of different NLRs, we actually find that there are people who have mutations in different NLRs who end up with different diseases that are called autoinflammatory diseases. So there are some people who have mutations in NLRs such that they have inflammation all the time, or really easily. And in most cases, what's happening is that people just have random fevers all the time. There's actually one of them on here, and I'm trying to remember how it is described here. So one of them is FCAS. I think familial Mediterranean fever might be the other one, but I'm not sure, because a couple of these are named not with a name I usually know, where you actually get inflammation from being too cold or too hot. Like a hot shower will give you inflammation in some of these cases. And all of these are because your NLRs are working overtime and making way too much of our inflammatory cytokines. And so these are some particularly important diseases. This is a very old and very out-of-date slide that shows you some diseases, some human diseases, where messed up inflammasome signaling has been seen, either because we can link it to genetic mutations, we can use a drug that blocks the inflammasome and see an effect in patients, or we can see animal models. And so what you can notice is, based on this, the inflammasome has been shown to be involved in those autoinflammatory diseases, which are in the middle, but also IBD, diabetes, hypertension, Alzheimer's, atherosclerosis, allergic dermatitis, type 1 diabetes, MS, lupus, vitiligo, Addison, celiac, rheumatoid. Basically, if you can think of it, in a lot of cases, the inflammasome has been linked to that disease state. And so this is one of the reasons why the inflammasome and some of these other PRRs and innate immune signaling proteins are some of the hottest proteins and targets in the pharmaceutical industry. The other group of PRRs that I want to tell you about are the ALRs. The ALRs are near and dear to my heart, since they're what I work on in my lab. One of the things that's pretty cool about them is that they are in either the cytoplasm or the nucleus. So once we learned that these could be in the nucleus, we realized that every single part of the cell was being checked for microbes. So that was pretty cool. It was like the last place that microbes could hide. No, they can't. There's a receptor there. And they tend to bind to DNA, which is, of course, a nucleic acid. So if you were going to guess, you were going to predict, what kind of transcription factor might the ALRs be related to? Yeah, Jay? IRF. Because, again, nucleic acid sort of ties into the antiviral response. In humans, there are 4 members of this family, the 4 of which are shown here. The first one described was called AIM2. So that's why they're ALRs, is the AIM2-like receptors. But there are a few others. They all bind to DNA and have a domain that allows for a protein-protein interaction. Different organisms have super different numbers of ALRs. So humans have 4, but like mice have 13, and bats have 0, and cows have 1. It's super weird. I think it's fascinating. Love it. But in any case, we've got these important DNA sensing proteins, and they have 2 different processes by which they can signal. One of them is that they can signal through a process that, oh my gosh, turns on IRF3. This shows them doing a little IRF and a little NF-kappa B, and that's really true. I've been oversimplifying to say it's always one or the other. In reality, it's a little both. But they do turn on IRF signaling. They do that downstream of a molecule called STING. I mention STING only because, again, in terms of targets in the pharmaceutical industry, STING is one of the absolute hottest. Every time I tell anyone that my lab does anything related to STING, they're like, I'm like, yeah, the stuff we're doing is not what you want, but that's cool. It's the stimulator of interferon genes. So that can show you that it's related to IRFs and interferon. Alternatively, ALRs also can make an inflammasome and lead to inflammasome signaling like pyroptosis or cytokine processing. And ALRs always are sensing DNA. Asterisk. That's okay. There's one other protein that I want to mention. This is yet another PRR, and it is called CGAS. You can see CGAS here. CGAS, just like the ALRs, is binding to DNA. CGAS has enzymatic activity, so it can make this small molecule product. It can signal through that same protein STING, and oh my gosh, lead to IRF and interferon production. CGAS is present in the cytoplasm, sensing DNA in the cytoplasm. So I mention it here because you can see similarities to the ALRs in that we're seeing some cytosolic sensing of DNA, signaling to IRF, making interferons, even using some of the same intermediates, STING. And so this is another way that you can see, here's CGAS, it makes its small molecule, we get some signaling. In reality, yes, it does do a little of both, IRF and NF-kappa B, and we get our defense. So in past years, this has been the end of what I have said about innate immunity. And we have then transitioned to adaptive. But I'm not going to transition to adaptive right now. I'm going to do that on Wednesday. And I'm going to just tell you a few other things that I find interesting, if nothing else. So we're going to be moving into talking about adaptive immunity next. If you look at the schedule, we are going to be talking about adaptive immunity and how adaptive immunity works for quite a while of this semester. Most of the time when people think about immunology and important things in immunology, their brain goes straight to adaptive immunity. They're like, yeah, that innate stuff, whatever. But adaptive immunity is only present in jawed vertebrates. Jawed fish, starting with sharks, as well as amphibians, reptiles, birds, and mammals, are the only ones that have adaptive immune responses. Fish that don't have jaws, as well as any other kind of animal, live perfectly well without an adaptive immune response. Some of this has to do with the evolution of proteins that we'll talk about next week. We can even look more broadly. Here you can see invertebrates and our vertebrates. Yes, they do have jawless fish here. Jawless fish are a little complicated in their immune responses. I'm going to kind of ignore them. We've also got plants. And again, all of our invertebrates, our jawless fish and our plants have survived billions of years with no antibodies, no lymphocytes, no aspect of adaptive immunity. So many organisms live perfectly fine lives without this layer at all. And so even though we're going to spend a lot of time talking about the adaptive immune response, the vertebrate adaptive immune response, the human adaptive immune response, know that evolutionarily it's only one option. In a lot of cases, phagocytosis and antimicrobial peptides and PRRs do it all. in those different organisms. Just to give you a couple of examples, this is a figure that shows some details of the innate, or of the immune response in plants. And so we've got a plant cell here. If you just generally look at this plant cell, if you didn't know and just looked at this image quickly, what might you notice or what might you think about this image? Does it look similar to anything else you've seen today? Does it kind of, yeah? What were you going to say? It looks like, I was showing you like a toll-like receptor figure. It looks really similar to the toll-like receptor figures. In fact, the proteins that plants use are structurally really similar to toll-like receptors. And what the plant tends to do is if the plant has, say, one leaf that's infected, that leaf will start to make small molecules that then they send to the other leaves. So the other leaves can do things like turn on antimicrobial activity or turn on immune-related genes. Does this sound in any way familiar? What kind of things does this sound like? Yeah. Right, like remember when we talked about the antiviral response? The infected cell says, yeah, I'm a goner, but I'm going to send interferon to warn my friends. It's the same thing happening in the plant where one leaf will actually send out small molecules to others. It's also actually really similar to pyroptosis. When the cell explodes, small molecules are coming out that go to the neighboring cells. So it's the same kind of themes that are everything that's needed for immunity in these other cells. In many of our organisms that are not vertebrates, we see other types of immune responses, and most of them involve proteins interacting with nucleic acid. And in fact, many of those proteins even have similarities to proteins that we know of in our immune responses. So it's a lot of the same themes, no matter which organism's immunology you're thinking about. Just to give you an example, I told you about C-gas. Some researchers did some work looking at C-gas, and what they found was that Drosophila, who is not a jawed vertebrate, got an invertebrate here, Drosophila basically had receptors that looked like C-gas, and they made a response that looked just like the C-gas response. And they were like, oh crap, if we actually look outside of humans, there's a whole family of C-gas-like receptors, CLRs, instead of just the one. So when we think of just humans, we're like, oh, there's just one. It's C-gas. But if we actually look broadly, we see that these are part of a family too, the C-gas-like receptors. And if we look even more broadly, we find first, yes, there is a whole family of C-gas-like proteins. Those proteins are in Drosophila. Those proteins are even in bacteria, helping bacteria respond to bacteriophage, viruses that infect bacteria. And so we can even see some of these immune responses that are listed here are actually types of immune responses that bacteria use to get rid of phages, the most famous of which is CRISPR. And so all of these are actually really variations on the same themes. So even though for pretty much the rest of the semester, we're going to be talking about the vertebrate immune system, and we're going to take sort of this deep dive into adaptive immunity, which is only in vertebrates. Everything that I've told you about innate immunity is relevant across the tree of life to pretty much every organism. There are a very small number where it may be irrelevant, which is fascinating also, but I don't get to go there. And hopefully it also helps you realize how important innate immunity is, because a whole lot of organisms, probably the majority of organisms on Earth, live totally happily ever after with just innate immunity and with no adaptive immunity. So don't sell innate immunity short. Okay, so the other story that I want to tell you is a story about some data from a paper. Because I think that the data from this paper illustrates some important concepts. Even though when I was originally thinking about having you read this paper, and they try to do 1,000 things and like put the kitchen sink in this paper, and so I didn't find it super readable when I went back and looked at it, but I think I can explain the key parts of the point of this paper. So in this figure, which is sort of the key figure we're going to be looking at, what these scientists have done is they've taken some cells and they've infected them with viruses. In this case, they've used 2 different viruses, either HPIV3 or RSV. Those are 2 different common respiratory viruses, parainfluenza and respiratory syncytial virus. So just general respiratory viruses. And what they're examining here is the transcription of thousands of different genes in those cells. Each one of these little lines is one gene. So there's like 20,000 genes across here as little tiny lines. If the gene got turned on when the virus infected the cells, then you get more of a red color. If the gene got turned off, you get more of a blue color. So like this gene kind of got turned off with RSV. These genes got turned on a lot with both viruses. So to simplify our lives, I put little labels on here instead of you trying to look at the weird way they labeled it in the paper. They're not looking at every gene in the genome here. They're just looking at some that are related to the immune response. But this area sort of is where there are some type 1 interferon genes. So you can see that when cells get infected with either of these viruses, type 1 interferon gets turned on. You get some red for type 1 interferon. These are also some things that are kind of related to type 1 interferon. Very red, very much turned on. These are ISGs, also related to type 1 interferon, turned on. This area is where genes related to NF-kappa B, transcription factor, are found. And so what I want you to see here is that while I have sort of been telling you, we make this distinction of it being one or the other, in the case of these 2 viruses, we're actually seeing both of them being turned on. And that can be important for sort of the immune system sort of having overall global homeostasis and balance. And so you can see that in these data. But when these authors looked at some other viruses, they look at SARS-CoV-1, influenza, or MERS, they see something that looks different than kind of their normal control viruses. So if you look at this, what do you observe with these other viruses? You can just describe what you see. Michael.
I feel a lot less like activation or transcription of the responses.
Yeah, you got a lot of white here instead of red. These are all mostly white instead of red, mostly red. The genes aren't turned on when these viruses infect the cells like the other ones. And what we know in a lot of ways is that some viruses mess with the immune response. And these are examples of viruses that actually inhibit IRF and NF-kappa B. You might guess where this is going. given that this paper is from 2020, and it shows things like SARS-CoV-1, that sort of implies that there might be something related to it. Before 2020, it wasn't called SARS-CoV-1, it was just called SARS-CoV, because we didn't know there was another one. So you might guess what's down here. And I, again, am only showing you part of this figure because they do 1,000,001 things. And so this is what they see when they look at SARS-CoV-2 infection of cells. So what do you notice when you look at the SARS-CoV-2 infection of cells? Yeah, or me. So can you say it a little louder? Yeah, if we look at the ISGs, they're missing. If we look at the type 1 interferons, they're missing. If we look at this interferon-related stuff, missing. All of that kind of looks like these ones on top. But if we look at the NF-kappa B stuff, it's still there. That doesn't look like these other viruses. It turns out that SARS-CoV-2 seems to be unique compared to all of these other viruses that were studied, in that SARS-CoV-2 inhibits one side of this process, the IRF side, but leaves the NF-kappa B side intact. So I will just show you another piece of data from this same paper. So here they actually looked at the interferon levels in patients. And they saw that whether the people were infected or controlled, they didn't really have any interferon. I will tell you that you should take this with a grain of salt because it takes a lot of interferon to show up in this assay. And so one thing that we do know is that the COVID patients probably had a teensy bit more, but you couldn't really tell. So maybe in reality, they're like here instead of there. We just can't really see it in this assay. And then these are all the NF-kappa B related cytokines. And what you can see is, in a lot of cases, the COVID patients are making way more of them. So again, SARS-CoV-2 seems to be inhibiting IRF, but not NF-kappa B. You can see that this is kind of the general idea. With a common respiratory virus, you get both. With SARS-CoV-2, you get low amounts of interferon, but you still get all of the NF-kappa B cytokines. One thing that people have realized, and there's some cool studies, is that SARS-CoV-2 actually requires NF-kappa B for its replication. So it can't inhibit NF-kappa B, because then it couldn't reproduce. But what you can also see is that we're getting this super different pattern of cytokines with SARS-CoV-2 infection compared to with other viral infections. SARS-CoV-2 is giving us a totally different pattern of cytokines. That different pattern of cytokines is what leads to many of the symptoms we see in our SARS-CoV-2 patients. So many of the things like some of the weird inflammation they have in their lungs is because they're making NF-kappa B related cytokines, but not the IRF ones to balance things out. And so a lot of what we actually see in the clinic is because of this imbalance in these. We also know some things about how SARS-CoV-2 inhibits this innate immune response. So it has a number of different proteins, they're all shown here, that can inhibit different steps of the signaling to IRF and of the interferon signaling cascade. So we can actually identify which parts of the virus inhibit IRF specifically. One thing to note, because sometimes this comes up when I show this slide, so you can see here the different proteins that are doing the inhibition, ORF3b, ORF9b, NSP8, NSP9, NSP1, NSP13, ORF6, NSP16, etc. One protein name that you don't see there, one protein name that does not show up on this slide, is S for spike. Spike is the only part of the virus that's in the vaccine. So the vaccine doesn't mess with your immune response, doesn't mess with your interferon response, but the actual virus does. And so this weird imbalance doesn't happen with the vaccine response, but it does with the virus. And that's one reason why the responses to vaccine versus virus are very different. Type 1 interferon and COVID-19 are one of many examples of sort of this Goldilocks model. Where here, as I've told you, it's kind of a problem that we're inhibiting this response. There's another group who have actually looked at patients who get very severe COVID, and they found that many of those patients have mutations, so they can't respond to interferon. So there are many patients who are basically unable to make a good interferon response. So the little tiny bit that is made is super important. And if you can't make that little tiny bit, then you're more likely to get severe COVID. And so they've shown different mutations in some patients are associated with life-threatening disease. They also point out in this paper, they have this sentence, 3 risk factors for life-threatening COVID-19 have been identified: being male, being elderly, or having other medical conditions. Immunologists have actually described really well the fact that your ability to do this interferon transcription process, your ability to do this process goes away as you get older. So in fact, the reason why being elderly can be linked to more severe disease is because we don't have this interferon response. There are a few things going on in the being male part, but many of the really important genes that we've been talking about in the innate immune response are found on the X chromosome, which means that males only have 1 copy, females have 2. So it's easier for males to have a defective one because they only get 1 shot and get a good one. And so you tend to see more issues with some of these genes in males. And so in fact, the being male can also be related to having problems in this pathway because of potential mutations altering the ability for this pathway to happen. So what I hope you realize are ways that this process and this pathway that I've been telling you about are super important in aspects of immunology. And we are going to start thinking about adaptive immunity on Wednesday. And I will see you then.